Hello and welcome to this joint presentation by I2C Pharm and Aptar Pharma. Firstly, let me introduce myself. I am Rachael of I2C Pharm and I will shortly be introducing my colleague Ségolène of Aptar Pharma. We would like to start by saying we hope that you and your families are safe and well during these uncertain COVID times. We are very pleased that the DDL committee were able to provide us with a platform to present our research, and so we thank you in advance for taking the time to view this presentation, even if we cannot be physically with you in Edinburgh this time. Let's get straight into the subject of the presentation: a dispersible salbutamol sulphate tablet for an environmentally sustainable HFA 152A PMDI. In this short presentation, we will demonstrate the effective combination of I2C's RespiTab technology and Aptar's valve technology for PMDIs. We will show how our key capabilities and commitments to the environment, as well as inhaled therapy sustainability, can assist with the changing landscape of PMDI development. As with any drug delivery development, an innovation that promotes sustainability must ultimately consider the needs and benefits to the patient. Therefore, I2C and Aptar have decided to combine their expertise in the RespiTab formulation and devices to help simplify and accelerate drug development and to ultimately benefit patients and society. In the next slides, we are excited to present efficient, uniform, and stable aerosol performance results that demonstrate the benefits of this collaboration. Currently, there is a strong drive towards the use of alternative propellants in PMDIs as part of the sustainability challenge. History has shown us that switching propellants is not an easy process, as experienced during the transition from CFCs to HFAs. MDI development is a complex and long journey of many different considerations, such as manufacturing, formulation, and economic considerations. I2C and Aptar have completed this successful RespiTab study to demonstrate how the development process can be simplified and accelerated. We have assessed manufacturing processes, hardware compatibility, and formulation feasibility with small-scale and short-term stability studies. The result is a novel approach of I2C's RespiTab with Aptar's valve technology that unifies flexible PMDI manufacturing process of the dispersible tablet with a simplified propellant filling process. PMDI development is a complex arena with several interconnected disciplines that are perhaps not always connected. Add to this the imminent transition from currently used HFAs. HFA 152A is more environmentally friendly but has different physiochemical properties, a different formulation, and manufacturing challenges. Hence, the future PMDI drug development space looks even more complex. RespiTab is a progressive formulation as it is compatible with both 134A and 152A. The technology allows for a safe and simple manufacturing process despite flammability hazards, with elimination of the need for homogenization and pressure vessels. Unlike traditional PMDI manufacturing, RespiTab allows a scalable, independent process from pilot-scale clinical trials to commercial batches. Providing both economic and environmental benefits through reduced API and propellant losses, Respitab is a faster, more affordable process with less manufacturing equipment and greater suspension stability. Respitab begins as a powder formulation consisting of micronized salbutamol sulfate with excipients menthol and lactose, which are blended together by low shear mixing, followed by analysis of salbutamol sulfate for content uniformity. This powder is then pressed into single tablets using a tablet machine. The resulting tablets are then placed into plain canisters and crimped with an Aptar valve. The canister is then filled with 152A propellant, with the tablet disintegrating upon propellant addition, which is demonstrated in the video on the following slide. This video demonstrates the disintegration of the tablet upon addition of 152A propellant through an Aptar valve. At the end of the standard quarantine period, all API will be dispersed. My colleague Ségolène from Aptar Pharma will now present the findings of this study.
Thanks, Rachael, and hello everyone. In this study, the field PMDIs were characterized in terms of dose content uniformity and aerodynamic particle size distribution. DCU was evaluated with 10 doses through CanLife as per European Pharmacopoeia, while APSD testing was performed using a next-generation impactor. Both tests were carried out at begin, middle, and end of CanLife, at initial time point and after accelerated aging at 40 degrees 75% humidity in inverted position. As we can observe on this graph, emitted dose for DCU showed low variability through CanLife at initial time point, with all results within specifications. Moreover, results were found to be stable along accelerated aging after 3 months. Going to APSD results at T0, we could observe that the distribution within the NGI stages was similar at begin, middle, and end of CanLife, with consistent fine particle fraction through CanLife. When looking at APSD along accelerated aging, fine particle fraction was found to stabilize after 2 months, showing a similar profile in comparison with Ventolin product. I will now pass it on to Rachael for the conclusion.
Thank you, Ségolène. The data presented on Respitab PMDI using HFA 152A demonstrates an efficient aerosol highlighted by high aerosol characteristics of fine particle fraction and uniformity of delivered dose. In addition to Respitab's high-quality aerosol, the use of standard hardware components showed limited effect on the physical stability of the PMDI formulation over the 3 months. We have also demonstrated that Respitab is compatible with 152A, thus helping to improve environmental sustainability of MDI manufacturing. We would like to thank you again for taking the time to view this presentation. Please find contact details for both I2C Pharma Services and Aptar Pharma on the following slide, should you have any inquiries.